Hello friends and welcome to my channel Easy Life Science. Here I am talking about a major histocompatibility complex or a MHC molecule. So please subscribe my channel Easy Life Science, like my video and share it to your friends. So let's start. The concept of MHC molecule was given by those two scientists, that is Gorer and Snell, in 1930. At time of rejection of a graft, the two individuals, that means a donor and recipient, should have to match their MSC. Otherwise, the graft will be rejected. These MSC molecules are useful for a successful grafting, as well as the role of MSC molecule is in a immune system to give a response and to present the antigens. For this discovery, the scientist Snail got a Nobel Prize in 1980. In all vertebrates, there is a genetic region that has a major influence on a graft survival or a graft rejection. For this grafting, the donor and a recipient should have matched their MSC molecules. The MSC molecules are nothing but glycoproteins, or these are glycoprotein receptors which are present on a eukaryotic cell membrane. The genetic region which is responsible for this glycoprotein are termed as a major histocompatibility complex. Unlike a minor histocompatibility complex, the MHC products play an important role in antigen recognition by T cells. Means the MHC molecule playing two roles. One is that for a successful grafting and for a presentation of antigen to the T cells. These are the membrane glycoproteins or cell membrane glycoprotein present in a vertebrates. The products of MSC plays a fundamental role in regulating immune responses. For this, the MSC molecule or MSC glycoprotein receptors load the antigen within a cell and shoot to the other rest of the cell in a immune system, so that other rest of cell in a immune system like a B cell, T cell activate and leads to formation of antibody and regulators like a chemokine, so that the antigen will get destroyed. T cell must recognize an antigen as a complex with a MSC molecule. For this presentation of antigen, firstly the antigen should have to proceed by unfolding and a proteolytic digestion before it complex with a MSC molecule. Once the complex of a glycoprotein MSC molecule and a peptide antigen is formed on a cell surface or on a cell membrane, it remains stable for some time. It has a half-life of approximately 24 hours. Once again, here is the mention a role of MSC molecule. The MSC protein or MSC glycoprotein binds a small peptide antigen and presents this peptide antigen to the T cell or T cell antigen receptor. The allelic variation of MSC molecule is functionally reflected in the selection of peptide which can bind it. Now, the location and function of MSC region. The MSC molecules are coded by one portion of a DNA present on a chromosome number 6 and 15 in a human, and chromosome number 70 in a mice. In a human, the MHC molecules are also called as a human leukocyte antigen. The MHC genes are organized in regions encoding three classes of the molecule. Class first, the class first MHC molecules are the class first MHC molecule are represented by all the nucleated cells in a eukaryotic organism, and these are the surface receptor. These are nothing but glycoproteins, and the class first is made up of the two chains, the alpha chain and a beta 2 microglobulin chain. The alpha chain has three domains or three external domains, that is alpha 1, alpha 2 and alpha 3. It has a cytoplasmic domain or a transmembrane segment with a cytoplasmic tail. This is a transmembrane segment of the alpha chain, and this is a cytoplasmic tail of a alpha chain. It has three domains, alpha 1, alpha 2 and alpha 3, and this is connected to a beta 2 microglobulin. The homology between alpha 3 and beta 2 microglobulin and concentration of immunoglobulins. Here, in this glycoprotein or in this class first molecule, we can see for one a cleft that is called as a peptide binding cleft, and on this peptide binding cleft, the antigen is loaded and presented or showed to the another rest of cells of a immune system, and this alpha 3 domain is a domain to which the T cell receptor get bind, and these molecules are expressed by all the nucleated cells in a eukaryotic organism, and they shows a procedure antigen to the CD8 cells or a cytotoxic T cells. Then the structure of MHC protein. The class first molecule are made up of one heavy chain, that is a alpha chain, having a molecular weight 45 kilodalton, and one light chain called as a beta 2 microglobulin. It has a molecular weight 12 kd or a kilodalton, and these two chains, heavy and light chain, contribute to the overall structure of a MHC protein or MHC first molecule. The MHC first molecule mediates a immune response against endogenous antigens that are already found in a cell. Basically, these cells express the MHC molecule which are tumorous in nature or which are virally impacted. The MHC first molecule presents a peptide antigen of a size 8 to 10 amino acids, and this peptide antigen then recognized by a CD8 cell or a cytotoxic cell. MHC class first glycoprotein cell membrane receptor are found on all nucleated cells. The class first molecule binds peptide and forms a complex with peptide antigen and a MHC molecule, and they present the antigen to a CD8 cells or a T cytotoxic cell. Each type of a class or a class first molecule having a further three regions: in human A, B, C, and in mice K, D, L. The single nucleated cell expresses 10 raised to 5 copies of each class first molecule. Many different peptides will be expressed simultaneously on the surface of MHC first molecule, and the MHC first molecule processes the antigen by a endogenous pathway or a cytosolic pathway. Then the structure of a class second molecule, it contains two different polypeptide chains. One is the alpha chain and another one is the beta chain. The alpha chain having a molecular weight 33 kilodaltons, whereas the beta chain having a molecular weight 28 kilodalton. Each chain has two external domains, alpha 1, alpha 2, beta 1 and beta 2. It has a one cleft on which there is a loading of a peptide antigen. It is called a peptide binding cleft. The class second molecule is a heterodimer, dimer of a dimer. The alpha 2 is a site for a binding of a receptor or a T cell receptor. The MHC class 2 molecule mediates immune response against exogenous antigen, the antigen which is found outside the cell and processed within a cytoplasm. The MHC second glycoprotein cell membrane receptors bind with a peptide antigen of a size 13 to 80 amino acid residue, and this will be recognized by a T helper cells or a CD4 cells. The MHC second molecule or a protein is found on a cell like beta lymphocyte, macrophages, monocytes, dendritic cell and endothelial cells. They are represented by a antigen presenting cell or a APC. The macrophages, beta lymphocytes and dendritic cell are a proportional APC or a proportional antigen presenting cell. These APCs phagocytose and engulf the extracellular antigen into a cytoplasm. The class second molecule binds a peptide and forms a complex, and this complex is represented by or it is recognized by a CD4 cell or a T helper cell. It can bind a variety of peptides and it processes the antigen by a endocytic pathway. Now about a genetic cluster of the MHC molecule. For mouse, it is called as a H2 complex. For human, it is called a human leukocyte antigen complex. It has three types: first, third, and second. Region for first is a BCA, and the gene products are like HLA-B, HLA-C, and HLA-A for first molecule. For second, regions are DP, DQ, DR. Their gene products are DP alpha and beta, DQ alpha and beta, DR alpha and beta. The class third molecule in a human, it has a region C4, C2 and BF. Their products are tumor necrosis factor alpha and tumor necrosis factor beta with C proteins. Whereas in a mouse, it is called a H2 complex. It has three types. The HLA, human leukocyte antigen region, it is coded or it is present on chromosome number 6 or a short arm of a chromosome number 6. This is a class first, class second and class third. Gene map of a human leukocyte antigen. In mouse or in other species, each class of MHC is represented by more than one locus. In man, this is called as HLA, human leukocyte antigen. The class first loci are HLA-A, HLA-B, and HLA-C. Class second loci are HLA-DR, HLA-DQ, and HLA-DP. All MHC genes map within a single region of the chromosome, so the term is called as a complex. Now, some general properties of the MHC molecules. Each MHC molecule consists of an extracellular peptide bonding cleft or a group on which there is a binding of a processed antigen or processed peptide antigen. Then it has a transmembrane domain and a cytoplasmic domain. The features of class first and class second molecule, as these are the glycoproteins, they are made up of polypeptide chains. In a class first molecule, two chains are there. One is alpha 2 and another one is a beta 2 microglobulin. Whereas a class second molecule is made up of alpha and beta 2 chains. Alpha 1, alpha 2, beta 1 and beta 2, these are the domains of the polypeptide chain in a class second molecule. The co-receptor alpha 3 domain binds with a CD8 cell receptor, but in case of class second molecule, the beta 2 domain binds with a CD4 receptor. Source for peptide, the class first molecule, the peptide or antigen are processed within a cytosol, and here is the processing of endogenous antigen. So pathway is called a cytosolic pathway or a endogenous pathway, whereas the class second molecule processes the antigen which is outside, it is engulfed, so the pathway is called as a endocytic pathway or a lysosomal pathway. The peptide antigen bounds approximately of 8 to 10 amino acids in a class first molecule, whereas a class second molecule binds a peptide antigen of 10 to 13 amino acid residues. Nomenclature in human, it is the first molecule named as HLA-A, HLA-B and HLA-C. Mouse, H2K, H2D and H2L for first molecule. For second molecule in human, HLA-DR, HLA-DQ and DP. For mouse, HLA first, HLA first A and first E. The class first molecule is expressed in all nucleated cells, whereas class second molecule is expressed by the APC or antigen presenting cell like dendritic cell, macrophages and B cells. In addition to these two classes of a MHC molecule, third class is there, and it has a role in complement components, and it produces a tumor necrosis factor, that is alpha and beta. It produces heat shock proteins, and these are not a membrane protein or a membrane glycoprotein, and so they have no any role in antigen presentation. The heat shock proteins are highly conserved proteins that are produced by a cell in response to the at time of trace, at time of nutrient deprivation, oxygen radicals, and at time of viral infection. And these are linked to autoimmune diseases. So thank you for watching my video. Please subscribe my channel Easy Life Science, like my video and share it to your friends. Press the bell icon